I suppose it's day 2, eh? And the paper is on seed system. Eh? Dr. Shinedu Wabara. Okay, that's let's bring in Dr. Shinedu Wabara to come and present what he has for us today. Please, Dr. Shinedu Wabara, can you quickly come to the podium?
Good morning, all, and thank you for the opportunity to be here. So my presentation is on strengthening farmer and community managed seed systems. I think this is not a new topic for the House, but it's something that's become of interest in Nigeria. A lot of investment over the years have been done in the farmer seed system, trying to support. Yeah, so a lot of investment has been done in the past trying to support companies to produce seeds and sell to farmers. But over time, people have realized that the community seed system dominates the seed sector. About 90% of farmers still source their seeds from the farmer and community managed seed systems. So in recent times, there's been that new push, maybe like last year and this year, from governments and also from from donors to see how they can support this farmer and community managed system. So we did this paper as a way to continue the advocacy to say, okay, yes, does the system really need support? My presentations I will talk a bit about the system perspective, then some key reflections from the system, some challenges facing farmer community managed system, and then we did some strategic innovation pathways on how to improve the farmer community managed seed system. So, in essence, what do farmers want when it comes to seed? Farmers don't know about formal system, informal system, or whatever you call it. What farmers need is quality seed at the right quantity, at the right time, at the right place that is affordable. So if you bring that to them, farmers will buy it. So they don't care what name you call the seed system. So whichever seed system that can deliver to them sufficient quantity of seeds at the right time, at the right place, and affordable. And this has to be adapted to their local agroecology. Otherwise, they are not going to use it. So if you have this in mind, this explains to you why and where farmers source their seeds from. Then we have this two parallel system of seed system that we have. So you have the formal system and the informal system. Okay, so the informal system. So if you look at the formal system, there is also a link between the formal and informal system. If you look at the formal system, it starts from gene banks, where breeders then use to make new varieties. Then they do the seed production, seed quality control, distribution. It goes to farmers, and farmers either save their seeds, and also then it goes through a lot of different ways. They also barter the seed, they sell it in the informal market, they consume some of the seeds, but at the end also, gene banks assess this. So the outside is what we call the formal system, and within, the inside is what we call the informal system. But the thing is that this informal system continues to dominate how farmers assess seeds in the country. And in Nigeria, we used to look at it as a negative constraint to say, okay, why after all this investment are farmers still sourcing seeds from the open market? Why are they not sourcing it from the formal systems that we've been investing and trying to support? But if you look other countries we try to analyze, this is no different. In Netherlands, the sourcing of seed from the informal sector is about 25%. And Netherlands, we know, is a very highly developed market, but still, farmers access seeds from the informal market. In Germany, it's 50%. In Canada, it's 65%. In Greece, it's 90%. So in Nigeria, we should not be shy when we say, yes, the numbers are 90%, and say, oh, we need to do to reduce it more. We see that even in much developed markets, that it's the same thing. But what we need to do is to professionalize the informal market, so that when farmers go there to get seeds, what they get is also the right quality seeds. Yeah, then a bit of this, very much all of these linkages between the formal and informal, how, genetics happen. I'm not a doctor, so I won't present this, but very interesting for you to look at. Yeah. All of the different ways. And, the farmers are also very important in community seed banking, in protecting our varieties. As we continue to breed and bring in more global varieties into the country, what we lose is our local varieties for some of our crops. And we know that some of these crops, there are no international interest in them. There are no companies interested in breeding them. So what happens to those varieties? We continue to lose our local indigenous varieties that has been adapted to our ecology, that has been more nutritious to us. So we need to really support this system. We looked across what, the array of farmer systems that are available. You have the farmer safe system, you have the local market, you have the community seed banks, you have the local seed businesses, and local seed businesses are where we think is like the intermediary between the informal system and the formal system. And we think they are very important, particularly those, we call them community seed entrepreneurs. They've been, over the years, there's been, Nrcri has supported a lot of them, and I think we should be proud of that. And at this moment where there is a lot of interest from the donor community to support it, we need to blow our trumpet so that we can see a lot more support come to Nrcri to really intervene in this system. Then we have the national seed companies, the multinational companies, and also the closed-end value projects. So what drives companies to invest in the different seed systems? It's two. We looked at it at two variables. One of them is around, is it easy to create hybrids? So hybrids, what companies do is that they are out to make money. If a company can develop hybrid seeds easily, they will do that, because what that creates is that if you buy a hybrid seed, you know that when you plant it, over time, the yield starts to drop, which means the farmer has to come back to them to buy. So, and that's how they make their money. That's their business model. So that's one condition. If it's an open pollinated variety where once you plant it, you don't need to go back to those companies to buy, companies are not interested in investing in that. So which you see some root and tuber crops. Also multiplication rates also matter, right? If you see maize, if you plant a grain of maize seed, how much you get out of it is very high compared to cassava stem, which is maybe ×5. This also motivates why companies will decide to invest or not. So if you continue to ask companies to invest in some kinds of seed system, they will never do it, no matter how much support you do to them. They will get the donor funding, and once the donor project ends, they go back to doing what they do, their maize seeds and their vegetable seeds. Another function is, are farmers willing to come back to buy those seeds, even if it's not a hybrid? And we see something like tomato seeds, right? If it's difficult for farmers to recycle their seed, to save it and extract seeds from it, what you see is that farmers will always go back to the companies to buy, and then companies make money from that kinds of seed. So if you look at those conditions, you see we created 4 buckets. In the sectors like maize, you see it's very high for seed companies to intervene. So yes, we can support seed companies to intervene in that sector. But you look at sectors where it's very low, then there is no point, supporting seed companies. What we need is more local seed businesses to to improve that, and this includes rice and also cereals. We know we are with the Nrcri's mandate on farming system allows us to do a lot beyond just our roots and tuber crops. So we can also take advantage of that for the Southeast to say, okay, we've been able to succeed in supporting the farmer community managed systems for roots and tuber crop. We can scale to other crops like rice, cereals, pulses, potatoes, and all that. Yeah, and then you also see where it's almost, you know, impossible to have private companies supporting in that sector. Then you need donors to intervene there. So we looked at what are some of the challenges and what are some of the advantages of farmer managed systems. You know, locally available, adapted conditions. Those seeds that farmers sell are usually always locally adapted, you know, and they are usually crops that are consumed in those communities. So it's just easy. How do you help them to clean it and bring it back to them? And you make your market easier and faster. The other point is also community seed system networks. Already at the village and community, there is already existing networks how farmers sell their seed. You don't really need to disrupt this. You just need to help them improve on that system, you know. So it's not about trying to create new market. The market is already there. If you go to the local communities, they have a way. They know in the communities who has the best seeds for the different varieties. How do you then help that person to be a champion, an entrepreneur, to sell your own kinds of seed that you've improved and bred, and they become that? So we also see Gene Bank. Sorry, I think I'm going too fast. Okay, so also in terms of conservation, we need, like I've mentioned earlier, we need to support our farmers to conserve our seed. Otherwise, we are going to lose a lot of our biodiversity that has been there with us for many years. Yeah, so these are some of the things we think that can be done, and we looked at other countries where they've been doing this, you know, this diversity seed fair, where you allow the farmers to show the varieties that they already have, that they've continued to use and people like, and this can be a way to attract other interest in it. We also see a situation where communities have biodiversity register to keep track of those varieties and the knowledge of growing those varieties. We need to support these kinds of activities and seed system. Then also, you know, farmers also have block activities, diversity, where they are allowed to do their own demos with their own varieties, not the improved varieties. And these are like some of the countries where these kinds of operations have happened. So we can also emulate this. Yeah, community seed banks, very important. And then, of course, something that Nrcri has been doing very well, which is the tri-cut, together with IITA, is also a very important way to bring in farmers into the breeding system to understand what is being, to be part of the breeding process for new varieties. So we listed a bit of challenges of why in Nigeria there's been conversations around supporting the farmer community managed system, but it has not really translated to results. And why is delayed? Some of the reasons are delays and limitation in policy implementation. We have the policies, but are they implemented? Inadequate storage and seed losses, which is very important. Deficiency in sustainability and business-oriented approaches, because this has to make business sense for it to be sustainable. Weak integration between formal seed system and the farmer community managed seed system. And we think that the National Root Crop Research Institute should be that plug that connects the two systems, helping clean the farmer varieties and also helping introduce high-performing varieties into the farmer managed system. Also, institutional markets where you see international donors import seeds when there is a crisis, they bring in bags of loads of seeds into those communities. It also affects the farmer managed seed system. So in terms of, I think this is very worthy, but we developed some strategic innovation pathways, which we think are pathways if we want to improve the farmer system. And one of them is around achieving a network of local seed businesses. And yes, indeed, we need to create more local seed businesses in those communities. And we identified steps in which this can be done, assisting in partnership support. I won't bore you with this. We have the paper and also the slides. You can go and read them after. Also, assessing agrobite. So also the aim is to establish a network of farmer groups that conserve locally adapted crop varieties. We need to have, so beyond the business of selling seeds, how do we conserve our local varieties? That is something that we have to do. And the final SIP is around to transform from emergency seed aid to sustainable community and market-driven seed system. What we say here is that when there's an emergency, maybe flooding or crisis or whatever, and farmers lose their seeds, don't import seeds. Try to support farmers to be the ones to grow back that seed banks that they have. Yeah, so I think this is my presentation. Thank you, everybody. So back to you, Chairman, sir.
We know that these days, arable land is increasingly decreasing, while population is increasing. And agriculture is tending towards intensification to ensure that we produce for the increasing population. And one of the factors of ensuring that we have enhanced yield is to make sure that we have quality seed, that we get to these people that are really responsible. So that they can discuss the details of what is there. What is, do I go ahead? I thought, okay, to discuss this, the presentation, we have Dr. Godwin Asmoga. Dr. Godwin Asmoga, as one of the panelists to discuss this. The next person there is Dr. Mark Tukula. I hope he's here. Dr. Mark Tukula. And can they assemble and do something here for us? Where is Onyeka? Please, Onyeka, can I see you? You know what you used to do now. Okay. Chinedu, please. You'll be there to moderate the whole thing. Chika. Okay. Chika Anyim, please. Chika Anyim. This is Chika Anyim, Acting General Manager of Nrcri Budike Seed. Is she here? Is coming? Godwin Asmoga, 1. Chika, 2. You, Mark Kula, and Mercy. Sitting order. Please, you are moderating this section, please.
Thank you, Mr. Chairman, for handing me back the job of moderation. But I don't know if my panelists have a mic. Yeah. So give to Dr. Mercy first. Okay, Dr. Messi, I know you have so much experience supporting through Go Seed, the community seed entrepreneurs. So this question is just to ask, is there a business case for this? Does it make business sense to intervene in the farmer community managed seed system? And how can research institutes work together with farmer community system, to produce more seeds of the quality that farmers want? So does it make, is it business, is there a business case, and can research play a part in that? Thank you.
Thank you very much, moderator. Of course, the answer to that question is absolutely yes. There is a business case to it. I mean, if there is no business case to it, then what's the point of even dabbling into it? But that being said, if care is not taken, this can be misconstrued. So things have to be done right. And in your presentation, you talked a lot about how to get this engaged and all that. But the part you didn't talk about is the business case. I think that is a justification, or will I say motivation, and as well as the varieties, not just introducing them to the new variety, but what is the key selling point of a particular variety? We currently just released Biggie and Renewed Hope. What is in there that is different from other cassava? Then they can start making it. But if they don't work together with if the researchers don't work with the farmers may not know this on time. It might take them years of cultivation for them to say, okay, why not do this at the beginning? And you now tell them, in order for you to make this a formal business for you, this is it. We take the opportunity to also to educate them about the formal system, and how it stands to serve them a better purpose than what they were doing before. Making it formalized, you have better markets for your product, you have better ways of putting monetary value because you know the quality of what you are producing. We have technology that enable you to produce quality planting materials. We have the rapid multiplication technology. We have DNA fingerprinting studies that can say, okay, because these are root crops, it is different from grain and cereal crop when you can see what is in between and easily tell what the variety is. Unlike cassava, sometimes if you don't harvest, you don't even really know what is there. We really need to know the nitty-gritty, and that is where the bridging of the gap between the researchers and the farmers coming. So the researchers have to put in more effort, and it is only then we can translate all of this to monetary value or the business case that is here. I could go on and on, but for time I'll stop here. Thank you.
Thank you. Thank you so much, Dr. Messi. I think you make the point that yes, there's a business, but we need to go about it the right way. The farmers need to also understand some of the things that the researchers are doing and why they are breeding some varieties. So it's not just breeding and giving to the farmers, and then at the end they don't really. It takes them time to then understand the purpose why that varieties were bred. So yeah, thank you so much for that insight. So I'll go over to Chika. Chika, your experience running Nrcri seeds now for some time. What are the main challenges that community seed entrepreneurs face when they try to grow and sell quality seed of cassava? And I think recently you also added yam. What simple things can help them to do better?
Good morning, everyone. Thank you for the question. Firstly, it's important I introduce Nrcri seed. Nrcri seed is an early generation seed. It's a commercialized arm of the National Root Crop Research Institute. We specialize in selling early generation seed. Then coming to the question, we have a lot of challenges, but because of time factor, I'll try to be as brief as possible. Firstly, we have a limited access to quality planting materials. From the start, if you don't get quality planting materials, then what you're doing is nonsense. It makes no sense. So there's a need for these community seed entrepreneurs to get it right by getting quality seed materials. And also, they also have this problem of inadequate knowledge of agronomic services. When they don't know it well, they don't know the technical know-how, how to handle these planting materials, or the spacing, or weed management and pest control, they also get it wrong. There's also pest and diseases evasion. When there is pest and disease evasion in a farm, it will also affect the input of the stem. Some of them also have this problem of record keeping. When they don't record properly, they will not be able to ascertain if it's sustainable. And then it also hinder them from getting finance where necessary. Also, they also have lack of access to markets. When they don't have access to these markets, then they can't sell. Even when they produce a lot, they have this challenge of selling. Why? Because they don't have networking, they have poor networking, and also they don't have linkages to these farmers and processors. Then coming to the solution, it's not just enough to just give the challenges we need. There's also need for us to give them the solutions. Then when you talk of solution, that is why my sister here from IITA Go Seed is here, and also Moody KC. There is need for them to go to get these quality planting materials from the EGS company to enable them plant well, start well. Then also there's need for them to continue learning. There's need for training. They need to keep learning to enable them get it well. Also, I also talked about poor record keeping. There's need for them to maintain at least a small, simple notebook or even a mobile app to enable them track the sales. Okay, this is what I spent for maybe labor, input. This is what I was able to generate to enable them ascertain if this can be sustainable. If this can be sustainable for them to be able to scale up. Also, there's need for them to also establish demo. At least demo will enable them to know, okay, this particular variety is doing well. Okay, there's need for you to come and plant these particular varieties to enable you, to boost the confidence of the entrepreneur. And then that will also be able to enable them to sell well that particular variety. Then last but not the least is for them to be able to at least, nobody is an island of knowledge. They need to also form group or join cooperatives to enable them to enable them face the market, and then if there's need for maybe any finance or funding, that will give them an advantage to be able to form part of the benefits. Thank you.
Thank you, Chika. I think you also alluded to the point I wanted to ask Dr. Mark on the groups, forming a group like you said, but I will get to that. But I think one point that resonated very much with me is when you talked about the numbers, farmers not understanding or not understanding the numbers. So I have the same issues, with, my parents are farmers also, and they don't understand that they make loss every year. They do farming, but, every year you support them to farm, and at the end, when you ask them, because they don't do the numbers, they don't realize they're making a loss. So at a point, I have to say, okay, look, I will just give you the money, stop farming, because it's been making a lot of losses and it's on my part. So it's even better to just give them. But of course, people discourage me to say, look, they also do it as a way to get activities done. But I think indeed, farmers, we need to train them on doing the numbers. So if they can do the numbers, then they can understand the business and what they are doing wrong and what they need to do better. So Dr. Mark, I come to you on the question of them forming groups. We know that Nrcri research, you've supported services, you help farmer seed groups, the Community Seed Entrepreneur Association that has been formed. But how can the institution help with training them better on the storage, on things like understanding the numbers, assessing the seeds like Chika mentioned, for them to know how to access quality seeds? How can the Root Crop Research Institute, how can you help in that regard?
Yes, thank you very much, the moderator. It's quite exciting to talk about the seed system and our support and how we galvanize all the resources we have. Nrcri, you will agree with me that one thing that is a thumb up for us is the abundance of high quality human and material resources that are available to be deployed to reach out to farmers in achieving our mandate as an institute. On the course of the year, one of the things is that this institute has well-structured laboratories to start from the soil analysis to inform farmers of what they need and how to manage and maintain the soil for optimum productivity of the crops. And when it comes to the seed system, we have been actively engaged in mentorship and backstopping of the seed entrepreneurs that we have formed. We leverage on the activities of the Basics project, where we are very active in the Southeast and South-South zone, where we have the ACCEN, that is the Association of Cassava Seed Entrepreneurs Network, formed. And we have criteria that have been set up with which we recruit people and train them, and then we keep on visiting them, just like the train and visit system of extension is a model that we have also, pulled into the seed system. And along the line, we also go ahead to carry out virtual training quarterly that to inform farmers who are not able to attend our trainings, physically. These activities has brought about making 1 million of seed entrepreneurs from this in terms of financial capacity. I make it bold to tell you that last year, a lot of success stories came from the activities we engage in with our farmers, with people, selling in millions. In Abia State here, I can tell you that there were several farmers here who sold up to 5,000, 10,000 bundles of cassava last year from the activities of the ACCEN. And it is moving like that. And then we have the primary catchment areas. That snowballs into the secondary people who benefit from the activities of the primary seed entrepreneurs, and it continues to spiral like that. And so we have a team. The team goes on to train people, and we engage one, site selection, like at this time now, we're on air trying to talk about site selection and what you can do. Another thing is that the branding that we have done in terms of the cassava varieties that have been developed and released. When you see 871105 days, farmers find it very difficult to call that. You bring it down like you say fine face. Anybody knows what is fine face? They attribute it to this particular variety. You say 419, like we have TME 419. Farmers go directly to that. They know what it is. And then we have the game changer, the Baba 70. We saw all of them on display yesterday. These are the branding strategy that the Institute has brought on board to bring farmers to be able to appreciate the qualities inherent in the varieties that have been bred. And then based on that, we also try to standardize the price, working with the seed entrepreneurship network, so that every year people have an idea of what it takes and to be able to do this. Now, because just like your presentation, to move from the informal seed system to the formal seed system, there are certain criteria that even the farmers, the seed entrepreneurs must meet. One, you must have an Android phone. You must have adopted one variety, improved varieties. You must be willing to teach other people. You must be available to be able to be, your farm to be accessible to NRCri. We have to connect the farmers to NRCri for quality assurance so that your farm will be certified. You see, one of the problems we have with cassava is that it is not something you can look at and say this is variety. Even when it starts growing up to like a month or two, you begin to see if there is mixture. And any farm that there is a mixture, it does not pass certification. And what we identified and we try to inculcate into our farmers is that if you move from here to any farm around here, you will discover that more than 4 or 5 varieties are planted in the same plot. And we are busy engaging with farmers, educating them. Right now, our farmers and ourselves, we are very, very active on the social media, using the Facebook, the WhatsApp, and several outlets to be able to pass across information that can be helpful to the farmers. So having done all this, we also be able to connect the producers to the demand of cassava seed. This is one of the major problems we also identify on the course of our study, and as an institute is a support to the farmers, because in those days when there used to be glut, people will have cassava in abundance in one place, people are looking for it, they don't know where to go to buy it. So based on the capacity building, mentorship and backstopping activities of the institute, we are able to connect demand to supply. We're also working with the early generation seed. The CEO is here, and Umudike seeds. We go to Go Seed and then Umudike seeds to try to connect them to be able to get high quality planting material. You see, about 90% of our cassava farmers like to reuse what they have. Farmers save seed year after year. And from time to time, based on NRCri regulation, you need to change those materials after some 3 years. So we have to consistently engage with the farmers to carry out these, quality assurance strategies. Of course, our farmers have grown to the level that they can get LPO and supply cassava variety. With the Seed Act, people now register small cooperatives and so on, and we bring those cooperatives together, both women and youth cooperatives, and we engage with them. We have also integrated a lot of permanent actors into the seed business. Nowadays, we have gone to Akwa Ibom State, to Imo State, and even to Abia State. The Ministry of Agriculture is very active all across the states where we cover. And let it be known that our activity does not just center on these states I'm mentioning, because the seed entrepreneurs are also present in Benue, in Kogi State. We were given a mandate to do 5 states. We have 14 states now in our kitty that we are working with them. And so based on that, we are popularizing the quality assurance, the seed system, the formal seed system, and the ability to be able to make money. Because previously, people always go to look for seed, cassava stem cuttings. You say, give me, I like this your farm, give me 2 sticks. Give me that by borrowing and exchanging. But more value have been added to it in terms of commercialization. So that is what we are doing. If you want, I want to continue, it will also be on and on. But we can see trailer loads of, seed moving from various locations in Nigeria to different locations. As we sit here, a lot of requests come, our phones do not rest, and then Nrcri is active on that. Thank you very much.
Thank you, Dr. Mark. Thank you. Yeah. So I think really Nrcri is doing a lot in this space, but yeah, more still needs to be done. Please, can you help me pass the mic? Welcome, Dr. Asmuga. Yeah, I can see you were following us online actually while you're there. Okay, thanks. Okay, the question to you, sir, is around, I've known you for many years now. You've worked a lot with the Cassava Seed Entrepreneurs Association. We've had several together with you. We've had some conversation with them as an association. So why do you think supporting these farmer groups are important?
Yeah, thank you so much, Mr. Moderator. Sorry, I was following you online while driving. I was head up somewhere. The farmers are the end users of whatever we are doing, end users of a research. If our research output does not go down to the farmers, that means there is a gap somewhere. We need to do a lot to make sure the farmers get the product of a research. It is important to get these things to the farmers because they are the people who are using these technologies, the small-scale farmers, medium-scale farmers, even the large-scale processors. Everybody, remember the thing now is agricultural transformation. So we have to do everything possible to ensure that the farmers get the product of all this research. We have to link them up. If I may combine this with, the next aspect of it. For example, Dr. Mark Tukula has informed us of the cassava seed entrepreneurs we put in place. I was leading that component while here, and when I visited, I handed over to him. So a lot has been done, and we need to do more. National Root Crop Research Institute has to do more. The project, if it is still going to be, we have to do more. You have to mentor them. Mentor the cassava seed entrepreneurs and the other entrepreneurs. Very, very important. It's very important to mentor them. You organize, trainings, go to the field, make sure you follow them up and they are doing the right thing. It is also very important you provide advisory services. Training services, flyers, extension guides and bulletins in order to give them to learn the new things we are doing. And very important, we must collaborate with the NASC. Because whatever we are doing, if we don't collaborate with the NASC, I don't think. There's a law now that whatever seed the farmers are producing, you must avail National Seed Council the opportunity to see your farm. Make sure you are doing the right thing. You know, very, very important. Now we are doing virtually trainings quarterly. Then I hope Dr. Mark is still on. This training is very, very important because it's not every time you have the opportunity to bring all the farmers in one place. Like he has about, I don't know how many states we are supervising now, about 10 or so. There are some states in the north. What we usually do is to link them to, you know, the virtual points. So that there will be virtual training. They will get in touch and informed about the new technologies. Any topic that we are to treat, they are supposed to key in. Is it a pest and disease control? The agronomy of the crops? Market development? Even a seed certification. They will get these things if they are unable to gather at a place and be taught. Also, step-down trainings. Very, very important. Step down. You have to, after being trained, you train the ESCO and train the ASEN. And then you organize the training at the farmers level. You go to their localities and they train them so that they will have a distance. And of course, it's good. We aggregated the seed entrepreneurs into an association. Now we have an association, National Association of Cassava Seed Entrepreneurs. And we have the apex body, we have the others. So these people will now coordinate the activities of the seed entrepreneurs, which is very, very important to make sure they are doing the right thing. Market day promo. I hope you mention it in your presentation. Market day promo, you go to the market and exhibit what you are producing. You know, you exhibit that so that the people in the market everywhere, you know, showcase samples so that they will know what you are doing, okay, which is very, very important.
Thanks, bro.
Yeah, am I speaking so much? Yeah, of course, you have to also engage with permanent actors. Okay. It's not only, you engage with local government, the ADPs, and all those, the credit agencies. You key the seed entrepreneurs so that they will get assistance also. Thank you very much.
Thank you, sir. Thank you so much, sir. So we just have 10 minutes and we wanted to take one or two questions from the audience. I'm looking at Dr. Sam. One question is enough from the audience? Okay. Any question? Let me even ask, maybe, is there any question for the panelists?
I enjoyed your presentation. However, there's something that I wonder if we have really addressed. I'm Professor Ifoma Ije. One of the questions that often arise is the reservation the local farmers have about improved seeds. You tried to talk about getting them informed properly. But still, you find that even the media is demarketing improved seeds, both on the social media and the formal media. So I don't know what the seed team is doing to address it. And oftentimes, they take the GMO thing out of context, Monsanto scandals and all that. So you find that because of social media, the average farmer now, you know, gets all sorts of information. that can sometimes affect their adoption of these improved seeds. Thank you very much.
I think everyone can answer this, but to put my voice to it, I would say in the same vein, even though farmers have been exposed to wrong information about improved seed, misconstruing it with GMOs, I would say there's also the other side. There's also awareness, more awareness created about the differences, even though there are some political undertones as how to adopt this material. So we are continuing in this business, and that's why we are saying as researchers, we are now more farmer-based. We are doing more farmer-based research, farmer-based breeding. For instance, like we said, what we are now doing is called a demand-led breeding. When we go right into the farm, what do you need? This is the process as which we're using it to do. I mean, if we're doing the conventional breeding, this is more or less like natural occurrence. It's just that we just use our hands to manipulate the movement and all that. No foreign bodies and all of that. So it's just continuous information, continuous education. And the seed system is such a great channel to do that, because now we are now in the formalized sector. We have the actors in the formal, everyone is within a regulatory framework. So as a farmer, you are being told that if your seeds are not regulated, you are not going to be liable to sell seed. Then you are mandated to conform to what they are saying, but it's not in a bad way. You are just being enlightened, and now making those people as champions, like in these community-based farming systems we are talking about. You get your champions, the people who will speak the languages that other farmers will hear. So we have to take it from up to down and the same way back. So it's two ways, not one way. It's continuous. If I say it's going to stop today, I'll be lying. These things will always be there. But what is our trajectory? What are we doing? What effort? And I would say the researchers have been doing a lot in this phase. The media as well. We really need to, as researchers, we don't have to leave these things for them. We also have to come into that space. And I've been talking about, I think I was with Nrcri some time ago. I was like, we leave some of very, very important issues to like all these content creators, kit makers. These are people that are gaining serious traction in the society. Why not let us use them as the right tool as well? Let's get them engaged in what we are doing. Every sector, I mean, this is a Greek. It's not like every other sector. This is life. People must eat every day. So definitely what we are taking in is as important as the information we are getting. So we have to be very involved. Personally, yes, I'm doing my own. But people that are working with the farmers, educate them, make them champions in their different localities. And I believe that with that, we're going to move very fast. Thank you.
Okay, Dr. Mark, just 2 minutes.
Yeah, thank you, Dr. Mercy. And thank you very much, Prof. It's a topical issue that you have raised. Some time ago, we went to interface with a women group in Ohafia, and right in the hall where we were to carry out the training, the issue of the GMO and all the misinformation came up. And so, in order to address that, even as a team, we decided to start operating this way. One, advocacy. Secondly, we decided to start setting up demo plots within the catchment location of the permanent actors we are engaging. Right now we have demos in some local governments in like Akwa Ibom, like in Abia here, Imo, so that that place that is a place where people come regularly, they will see what we are doing, that it is exactly the practices that are being carried out on their farm. We now have field days in those places and then show them how to do it better. And then we also sometimes we have organized our CSEs into clusters across the states and at the different locations. So now, like in Abia here now, we have a whole lot of cluster, like in Amakama, where we have the CSEs, so many. In that cluster, we also put our demo in the midst of the farm of the CSEs where they are located. In Benue, it is like that. In Kogi, it's like that. In Akwa Ibom and all around, we do that. So that at the end of the day, we can now disabuse the mind of the people that there is nothing hidden about what we are doing. apart from just the GM, people also have all manner of misconception about rotting, about water, about so on. So we are going ahead now to promote the various qualities that each variety has that is different from another variety. We are even going ahead to make pamphlets around it so that you will know if you are going for, why is it that 80% of demand, like we are doing varietal penetration, diffusion of the varieties that we have information about it. Why is it that a whole lot of the requests that we have goes to TME 419? Why is this other one trailing? Then we begin to be able to educate the people that what you are looking for in TME 419, you can also get it in Dixon. You can also get it at some level in other varieties, and so that there can be balanced marketing. So we are doing a lot to try to bring about changes in the approach. Thank you.
Thank you so much. Yeah. So a round of applause for my panelists. Yeah. I'm not sure I'm allowed to take any other questions. So unfortunately, you can still meet the panelists afterwards and ask your questions. Thank you.
Yeah. Thank you so very much. Can we give another round of applause? I would want to say that you've tried to keep to time, though you exceeded by 5 minutes. Exceeded by 5 minutes. You only have 20 minutes to present these things. I hope you people are guided aright so that we can live here. With that, I think my job is done for now. The person that I'm standing in for is around. I can take my leave. Thank you very much.
Thank you so much, our standing in chairman that has done a wonderful work. As usual, all the panelists, the moderator, and the chairman will take a group photograph. Can we, cameraman, please, quickly. Chairman, sir, you want to take it standing, sitting? Chairman is coming now. You want to create, let's stand so that we can make it fast. Chairman, please, sir. Let chairman be in the middle. Chairman should be in the middle of the two beautiful ladies. Between the two ladies. Cameraman. So, acting ED, please, you do something for me, sir. Oh yeah, come. There's one you are going to take. Oh yeah, stay. Oh yeah, Director PME, join quickly. No. Okay. Have you taken it? Quick. Thank you. So, Acting ED, please, you will take panel members. You can leave. Take with Dr. Law. There's a reason for that. Yes. Dr. Law is a retired director. Go ahead, take the picture. He's a retired director, but when he was in service, by 7:30, 7:45, he's here. This morning, by 8:20, Dr. Law was here. Continue to be who you are. Thank you. Can we put our hands together? Thank you so much, sir. That was really, I met him, I said, this thing dey your blood. You know if he change. Thank you so much, sir. God bless you. I appreciate. Thank you, sir.
Ladies and gentlemen, at this point in time, we want to move over to the technical session, and we want to call up the chairperson for this session, and she is Professor Ifoma Irene J., Professor of Biochemistry, and a member of Board of Directors of Muayu Nigerian Limited. Prof, you're welcome. Please, I want to ask that you clap very well. Thank you so much. I want to hand over to the chairman, who will now direct the next procedure.
Thank you very, very much. I want to sincerely apologize for being late this morning. It's not in my character. I had some glitches. So we will be going to the technical session, but I don't know if we should allow the rapporteurs to... Do we give them a...
Yeah, yes.
They're still working, okay. So thank you very much. We'll be taking the reports from the various programs, and I think the first report is coming from... okay, we'll have a proposal presentation, then we'll have the questions. So the first will be coming from the product development program. I don't know if they're ready. Joe? Okay. Our chairman that just stepped out talked about time, so he was particular, so we'll follow that cue. You have 10 minutes.
Okay. Good morning, my chairman, the executive directors, distinguished guests, fellow researchers, good morning. So I wish to present at this moment the annual report from the biotechnology program. Next. Next, please. Okay, so this is my outline. Next. Okay, so this is our research. Okay, thank you. Okay, so these are research areas: molecular breeding, molecular diagnostics, genetic modification, tissue culture, and bioinformatics. So our annual report. So the first one is on exploring target tissues for us, exploring alternative target tissues for accelerated cassava genetic transformation. And the key objective here is control of agrobacterium overgrowth. So the point is that when it comes to genetic transformation for cassava, we normally use what is called the Fraba embryogenic callus, and it has a lot of limitations. Number one, it takes a long period of time, and number two is genotype dependent, restricting us to one particular type of genotype to work with. So we're looking at alternative methods, one of which is to use explants to derive transgenic materials. But one of the challenges we encounter was what we call agrobacterium overgrowth, so we now decided to look at how we can control this. So the objective is determine optimal antibiotic concentration that support regeneration while eliminating agrobacterium, and then evaluate the impact of the different antibiotics on cassava tissue regeneration. So the key achievement here is that from our research or from this experiment, we're able to control overgrowth of agrobacterium in alternative tissue. And the lesson learned here is that we can do cassava genetic transformation using other methods that are not dependent on the FEC. Okay, so what we looked at viability, we looked at vigor, biofilm formation, explant degeneration. We used two major antibiotics, the cefotaxime and carbanicillin, and the conclusion is that we could use moderate concentration of cefotaxime, 400 to 600 microgram per mil, to do effective control of agrobacterium overgrowth without impacting the vigor and viability of the explant, while for carbanicillin we can use as low as 125 milligram. Okay, so the next research is understanding the molecular mechanism of starch biosynthesis in cassava. So this work was done by a PhD student, and the objective of this research is to understand the mechanism of starch biosynthesis in cassava during tuberization. Okay, so from the results obtained, the transcriptomic analysis showed that there were about 175,808 genes that were associated in starch biosynthesis at 4 different time points. And out of these number of genes, 20,402 genes were found to be differentially expressed. That is, there are different expression at different levels. And out of these 20,000, 15 key genes were identified with cassava starch biosynthesis in cassava. And the main gene out of the 15 is the sucrose phosphate synthetase gene, which is the SPS. So the summary of this research is that this was done in 419 genotype, so, and which is high in dry matter content. So the story here is that having identified this SPS gene, we can overexpress this gene using biotechnology methods to improve dry matter content in other genotypes with less dry matter content. Okay, so the next work is also a PhD study that was done by Abba Simon. The first was done by Ruben Jane. And the objective here is to kind of identify structural and functional genes associated with dry matter content, knowing the fact that so many other factors could also attribute to DMC. So, but in this research, we concentrated more on this particular gene that is called the actin-related protein gene. So, this gene was sequenced, and genomic sequence was translated to protein, and the protein sequence was blasted to show the hits in different other organisms. And this is what this place is showing. And you could have other families, other crops that have the same gene, in which it has been validated that in these crops, that particular gene is responsible for DMC. Okay, so what was further done was to use computational biology to get an amino acid structure of this particular gene, and then a kind of dog it together with water and ligand molecule, and it shows this model. So what is showing here, like this is the water and the ligand that is associated in dry matter content. So what is showing here that for this particular variety that is high in dry matter content, you could see that the binding of the water molecule with amino acid is less, showing that it has less water. So what is the story here? What we're trying to say is that we could use this method to determine which genotypes that have the ability to have dry matter content and those that have low dry matter content. So that was the essence of this research. Sorry. Okay, so the next report here is screening of semi-autotrophic hydroponic, SAH for short, derived cassava varieties under different weather conditions for effective establishment. Now, what a kind of suggested this research is because we have farmers come to us and say, we want to take the SAH materials and we want to establish it directly in our field. What are the things that we need to put in place? So we needed to do this experiment to find out when is the best time to go to the field, what are the necessary things that are required. So that is the objective of this research, to look at the different times, seasons that we can plant this, because these are plantlets coming out from the lab. Establishment rates most times are being affected depending on when it was established. Okay, so So we looked at different period of different weather conditions. We looked at dry, early rainfall, peak rainfall, and late rainfall. And the result is showing, we looked at 5 genotypes. We used Dixon, Fine Face, Obasanjo, Sunshine, and TME 419. And we could see that the different genotypes behaved differently at different times. Some of them, like the ones that are linear, shows that they were stable over time, and they were the ones that we recommend for the farmers, that if you go with these varieties, they are well able to establish in the field and give good yield. So the conclusion of the matter is that weather conditions can influence SAH-derived cassava establishment, and we have better establishment when you plant at the peak of rainfall. And then we are saying that the SAH technology can actually be optimized as a viable propagation tool for cassava production when we consider all the necessary factors, including the weather. Okay, so the next research is also a PhD study that is looking at quality control and generation of self-botanical seeds for crosses using genomic selection tool. So the story here is that we used genomic selection to identify offsprings that have high starch content and high dry matter content. So the point here is that the phenotypic data of the parents, which are written over their TMS, were collected, and the offspring that resulted from the crosses were sequenced, and genomic data was derived. And using the combination of the phenotypic data and the genomic data, we're able to do a prediction. And the prediction here shows, so this from the predictions, this number of offspring were selected as outperforming the parents. And at this moment, these materials are being evaluated in the field. So what is the importance of this? You remember when you do crosses, you get thousands of offspring. But with genomic selection, it reduces the number of years that you're going to use for the evaluation of this offspring. Okay, so do I go ahead with the proposals? Okay, so for our proposals, we have proposals in the different areas: molecular biology, molecular diagnostics, genetic modification, tissue culture, and bioinformatics. So the first one here, we're looking at molecular characterization of new taro germplasm. And the researchers are there. The main target here is to determine the extent of genetic variation within and between taro populations. Then identify the unique genotype that we use for both breeding and conservation. And then as well, identify genetic markers that are linked to desirable traits like taro leaf blight disease, yield, and quality. So the expected outcome is that we'll be able to identify diverse gene pools for breeding. We will be able to link markers to traits like disease resistance and yield, and we'll be able to verify the taro accessions, cultivars, and land races. The second research, we'll be looking at the study on the genetic diversity of taro leaf blight, Phytophthora colocasia, among Nrcri germplasm, and those are the researchers over there. And the objective is to determine the effect of genotype on disease severity. Then we want to look at characterization of the causative agents among Nrcri germplasm, and then with the information acquired, we'll be able to develop a mitigating strategy for the control. So the next research proposal is evaluation of biological activities of Phytum oligodendron on rhizome soft rot disease of ginger in Kaduna State. This is a PhD study, and the target is to isolate and characterize the potential beneficial microagents microbial agent for controlling rhizome soft rot disease in ginger, to assess the in vitro antagonistic activity of beneficial microagents against the pathogen, and then evaluate the efficacy of the biocontrol strategy. So the next proposal is induction of resistance to cocoyam root rot disease through ethylene metanosulfate mutagenesis and identification of resistant gene. This is another MSc. This is an MSc project, and the target is to induce genetic variation using EMS to evaluate the phenotypic variations in morphological, physiological, and agronomic terms, and then to identify potential mutants with improved disease resistance. So those are the expected outcome. Then next is callus induction and optimization of meristem regeneration of taro towards gene editing studies. So, and the objective is to develop an efficient protocol for callus induction and meristem optimization of taro cultivars, done to evaluate the genetic stability and yield performance of regenerated taro plants. Okay, next is green synthesis of metallic nanoparticles using extracts of ginger plants for use in agrobacterium-mediated cassava transformation. This is also a PhD, part of a PhD project. So we heard about nanoparticles. Our keynote speaker talked about nanoparticles. And we want to use ginger extract here as a nanoparticle to help improve the agrobacterium-mediated transformation. The point is that agrobacterium-mediated transformation efficiency is quite low, but we are kind of exploring that when we use ginger extract nanoparticles, that we could improve the efficiency of agrobacterium-mediated transformation. Okay. All right, so next we are looking at genomic insight into transformation compatibility. Here we are going to use bioinformatic tools to understand, for example, I do cassava transformation, but you discover that it's not all genotypes that are amenable to transformation, and this is usually a big problem. But there are some that are amenable, some are resistant. So we want to use bioinformatic tools to evaluate those ones that are amenable and those ones that are not amenable, so that we can find out what is the underlying principle. And when we do that, it will help us to improve the system. So next is molecular docking of key metabolites against disease-causing pathogens in root and tuber crops. This is also a bioinformatic research that we're going to do to look at the key metabolites that are associated in disease-related relationship, and kind of use bioinformatic tools to form a module to look at, okay, what is the key role of these metabolites, and how can we tweak these metabolites to a kind of improve our understanding of the disease mechanism. And next is flux balance analysis of primary and secondary metabolites influencing yield in root and tuber crop. This is also bioinformatics. So we want to kind of use bioinformatic tools to reconstruct and refine genomic scale metabolic model for root and tuber crops. What we're saying is that, for example, when you come in gene expression, especially a trait that is a kind of a trait that is influenced by multiple genes. Normally, there is one gene out of the many genes that when it is regulated, it induces the regulation of the other genes. So we want to look at the model and see if we can manipulate this gene either for our benefit by overexpressing or by knocking it out, depending on what the objective is. Okay, and lastly, we're looking at confined field trial of gene-edited cassava resistant to cassava bacterial blight disease. So presently now, we have gene-edited cassava in our lab, and our institute in Nigeria is the first to get permits to carry out gene-edited research in the country. So we have these materials available at the moment, and what we're trying to do is to determine the expression of the intended traits in the field, then evaluate its impact on CBB resistance, and then determine the most impactful strategy, because there are two strategies that are used in developing the resistant materials. Thank you.
Thank you very much, Dr. Puano. I kept waiting to hear what you want to do with cassava, and there was tyro till we got to field trials. So well, we've seen the ongoing work and then the proposed study. And you appear to have a lot of dry lab for the coming year.
We actually have a lot of ongoing.
Okay,
yes.
So you want to finish what you have.
Exactly.
That's why I added.
Exactly.
Well, anyway, most of the dry lab you are proposing will also answer questions to the ongoing. That will be quite okay. So thank you very much. Please, can we... time for the answers. Thank you very much. So Sam, I think you take it on from there and let's have the questions. So do we have questions for her? We have a hand up there. Another hand beside him. Oh no, that's not balanced, but it's okay. Let's take them. Okay, one behind. In that case, each question will be less than 1 minute, so that, please, we'll just note the questions so that you take them all together once. Thank you very much. You have a pen?
Yes.
I can donate mine.
Okay.
Okay, thank you.
I'm Chief Okafuzu Gochukwu from the National Yam Information Research Center, Ibuku. My question goes, in 2009, our people, when the Nrcri came to Ibuku for their lecture, our people asked, what can they do about this cocoyam, the taro blight disease? How can they handle it? And since today, this institute has not given us the real way of how that disease can be handled. And you brought it up now. I want to know more about it. Thank you.
Thank you so very much for the opportunity given me. It's not a question as such. This issue had come up here some time ago. A topic is raised, and you have researchers under that. At the end of it, you mentioned that it's a somebody's master's job or PhD job. How do you marry that with so many authors under that? Thank you.
Do I ask my own question? Please, you talked about enhanced dry matter. enhanced dry matter in cassava. I recall that in 2006, when we went on a survey for the collection of farmers' preferred cassava variety in the country, I anchored Lagos and Oyo State. In Oyo State, we went to the farmers and there was one cassava variety. I think they call it Ogondo or something like that. That cassava variety, they said it yields very well, but they had it at the border, they never put it on their farm, but at the border rows. I said, why are you not having it on the farm? They said it's full of water. That's why they're not, but the yield is fantastic, very fantastic. And I told them, that's why we have come for this survey. I'm in biotechnology. We will see what we can do to enhance that dry matter in that your cassava so that you can continue to grow it and have
fresh enough. And it was also not able to come back to its shape after being touched. For the storage days, we found out that the days of storage also had significant impact on the hardness of the materials. The day 5 materials were also by that time hard, and it's as a result of changes in the structure of the starch. And if you look at our storage condition, you find out that those that were stored in the fridge, they were harder. That's because when you keep it at the room temperature, the gluten tends to relax more slowly, and then it will take time to become soft. But when you keep it in the fridge tends to stop it automatically from doing that. So for storage condition, we found out that keeping it at ambient temperature gives you bread that is soft and able to keep more. Now we took it a step further to check for the total microbial viable count. That was supposed to be a storage study. How long can this bread keep? We are selling it to people, we are promoting it. How long can it keep on the shelf and in the fridge? From our study, we found out that our sweet potato bread can last across the genotypes for 3 days comfortably. But we've been able to identify some of them that can stay up to 5 days without having any growth on them. That is the okunkum and the tea purple. And for the breeders, I think this is a good study for you to be able to check what is keeping these things stable and try to cross it with the other ones. The tea purple is not giving you good textural properties, it's not giving you good appealing properties, but is keeping very well. So this is it. So our achievements this year is to, one of the things we achieved was that the M color, the Mother Delights cross color can be considered to be one of the most appealing to the consumers. Okunkum's sweet potato bread is likely to be the most acceptable to consumers due to its softer and texture and lower chewiness values. The tea purple is more shelf stable, and this suggests potential antimicrobial traits. Lesson learned. Genotype selection is crucial for optimizing color, texture, and shelf life in bread. And storage condition critically affects bread quality. Since refrigeration helps to maintain color and texture, we then suggest that it should be kept under that condition.
Thank you very, very much. I hope you are done. You still have another product. You will need to.
First with these ones now.
You will need to, but just on a lighter note, the tea purple has far higher antioxidants. So naturally, it should confer antimicrobial properties. But it has the disadvantage that the starch is more highly branched. So it's for the breeding people and the biotech people to look at the down-regulating some of the starch synthase activity.
So our next activity last year was also the oat bread for using sweet potato flour and the white and the wheat. And from the study, we were able to understand that we tried 3 different ratio substitution, 20, 50, and 70. And from the study, we found out that the ones that have 70% substitution has better starch quality. It has better starch quality, lower sugar content, and more of the amylose. So and is competing more favorably with our control. I'm rushing, ma. So we took it a step further to check for the blood glucose glycemic index. This study came up as a request from one of our researchers that is diabetic. So he wanted us to add some fortification with the orange flesh sweet potato and also try to give him the oat bread that he's buying from the market. From our study, we got an ethical clearance from the Federal Medical Center, Umuahia, before we conducted this study. And from our study, we found out that 70% substitution with 10% fortification of the OFS puree, we could get a GI that is medium compared to what is in the market and compared to the other substitution ratio. So I will get to the next study. Here we are looking at also making instant yam flour from alata accession. You don't use alata to make poundo yam. So we are trying to develop a recipe that is also suitable for these vulnerable people, and we're able to achieve it. This is our flow chart. I've removed the things so that we can be able to patent this material. From our study, we found out that the genotype TDA 1817 1049 has good amylose and amylopectin ratio. And from literature, this is what is needed for you to be able to get a dough that is cohesive, that is moldable, a dough that has good dispersibility, so that when you are reconstituting it, it can stay. It's not going to be adhesive to the machine. It's not going to be sticky to the hand. So we took it a step further and then checked the textural properties just to confirm what I have said here. And you find out that the same genotype has moderately high hardness compared to the other genotypes. The adhesiveness is also lower. It's a negative value, and the cohesiveness is also very good. We also checked the effects of the two different methods of processing that we use, and we found out that using blanching method tends to give you a better product compared to boiling of your jam genotypes for the processing of the product. So what is our lessons learned? We've been able to learn that there is genetic differences in the physicochemical properties of the flour, as well as the dough textural properties, and that blanching yields better, cohesive, and resilient dough after reconstitution of the flour. The next study is a gender study that was carried out by the authors in the slides, and it was supposed, and we're looking at the gender analysis in processing of some selected roots and tuber crops, ginger and turmeric in Abia State. From the study, we looked at different activities, which include the gathering, washing, splitting, milling, packaging, gathering, washing, and all. And we found out we wanted to monitor the roles of gender in these activities. And we found out that across board, all the things that have to do with manual are more of the male roles, while the lesser ones are more, females are more involved. We looked at the factors influencing the male participation, and we were able to find out that the household size, the age, credit facilities, and membership to cooperative is a very big factor that influences participation of males in ginger processing within Abia State. For the females, we also found out that their marital status, the household size, the age, and processing experience, and the membership to associations also influence their participation in processing of these two opportunity crops. So, achievements, we found out that gender roles, there is a clear identification of gender roles, and that it involves, and the gender determinants in the and statistical validation of the determinants were done. The lesson learned is that women dominate gathering, washing, and all other small unit operation, while the men are more involved in things that are more stressful than males. Then both gender face challenges such as lack of raw material, milling machine, and high cost of processing factors. The next study we studied the characteristics of the culinary properties of some selected sweet potato genotypes. We did a dry matter analysis, and we found out that the materials differed with respect to dry matter. This shows us that if you have a material here that has low dry matter, it is a very suitable candidate for making an OFSP drink, while the ones that have high dry matter content can be channeled into baking and boiling. We checked the oil absorption capacity, and we found out that the variety also plays a key role in this particular functional property. So we have those that, the ones with high oil absorption will suck oil when you are frying them. So we do not suggest that anybody processing sweet potato chips should collect Alicia or Victoria as a material to use for processing the chips. So we looked at water absorption and cooking time. From the study, we found out from lessons learned that the varieties differ significantly in culinary characteristics affecting their end use, and that the high dry matter contents are preferred for frying and baking due to better texture retentions, and the varieties with shorter cooking times are more desirable both for consumers and the food industry. The other study we did here was assessing processing method and glycemic index of orange flesh sweet potato for improved dietary consideration. Here we also went and got an ethical clearance because we took the fasting blood sugar of some people to be able to get the data from this study. And from our study, we boiled and we juiced. So from this study, we found out that across the day, between the two methods of processing, that they all have high glycemic index, which means if you have this spike of sugar, we do not advise you to start eating boiled potato or to start drinking the juice, because we still need to work on it to be able to find a way to ensure that you don't get into crisis after consuming them. The last one is comparison of different multivariate selection indices of selection of superior cassava genotypes with good food quality traits. This is from a breeder who looked at the different food quality traits that we measure during processing of our cassava. This is not a normal thing. Breeders normally concentrate on agronomy and disease, but we're integrating food quality. So he wanted to find out which method, which index is best to use to select materials with good food quality traits. He considered dry matter, starch, peel loss, Gary yield, and the Retin index. And he used 5 different selection indices. From his study and from our study and result, we found out that the material G8 was the best for all the traits considered, and that the different indices are different, have different ways of selecting materials. But when you combine all of them together, these genotypes that are being bred stand a better chance of being selected properly. So this is the ranking table that I just mentioned now and the models he used. So the lesson learned is that considering multiple traits simultaneously provides a more accurate and efficient selection framework. And the selection index has unique strengths. Combining method enhances confidence in identifying top performing genotypes. Proper standardization, e.g. Z-scores and thoughtful trait weighing, improve selection precision and relevance to breeding objectives. Thank you.
Thank you very much. Well, I think we can forgive the extra 5 minutes, nearly that. So that was a good presentation. I must commend the Institute. All the programs appear to have had too many projects within the year, and all well achieved. So we will look at your proposal presentation then for the coming year.
Okay, for this year, we have the first proposal is optimizing conditions of taro genotypes developed, storage conditions of taro genotypes developed, implication for seed quality, culinary, and texture properties. We want to do this based on what we met in the ban last year. And we found out that our current ban is maybe being influenced by climate change. So we want to revisit storage of taro within our agroecology. We also want to do, the second proposal will be evaluation of cookies from black seed or FSP wheat blend as a potential functional food for obesity control. This black seed is going to serve as our functional seed. So we'll include it and see how best we can use this to check obesity using our mandate crops, products from our mandate crops. The third one is quality evaluation of flakes from white and purple taro blended with fermented okara, that is soya bean residue. And the fourth one will cover or address impact of different heat treatments and time on glycemic index and antioxidant properties of three genotypes of orange flesh sweet potato. The objectives will be to determine the physicochemical properties of some newly developed sweet potato genotypes, to determine the processing characteristics of the newly processed sweet potato genotypes. The fifth one will be quality trait profiling of potato varieties in JAWS. We will want to identify potato varieties with superior quality traits for cultivation and consumption, to evaluate the nutritional composition of potato varieties in JAWS, and we'll be concentrating on these chemical qualities, to analyze the sensory qualities of potatoes, which include their taste, texture, and cooking qualities. The sixth one will be design and development and evaluation of dried turmeric rhizome process polishing machine with grinder. And what do we intend to do? We want to design and develop a dual-purpose engine-powered machine for polishing and grinding turmeric rhizomes. We want to develop an efficient machine that can be used to produce the turmeric powder. We want to evaluate the performance of the developed machine, and we want to enhance the production and quality of turmeric powder. The seventh one will be addressing the descriptive sensory evaluation of extruded breakfast flakes produced from orange flesh sweet potato and Tania blends for consumer acceptability. The objectives are on the board. The eighth one will involve evaluation of proximate chemical properties and microbial load of extruded breakfast flakes produced from orange flesh sweet potato and Tania pulp. The ninth one will address characterization of flour from underutilized yam species, that is the adu. That is what the particular one we want to use for physicochemical properties, nutritional composition, and for potential food application. The 10th one will be functional and nutritional evaluation of sweet potato crackers and the impact of orange and white flesh varieties on the quality and acceptability. The objectives are stated here. The 11th one will address production and nutritional benefits of ginger, turmeric tea blends with cloves and cinnamon spices. And these are the objectives. The 12th one will involve exploring different approaches to reducing postprandial glycemic impact of orange flesh sweet potato food products, strategies for diabetic consumers. That is from the previous work we did. And the 13th one will be study on the flavor and sensory profile of orange flesh sweet potato using probiotic bacteria. This is a PhD work. And the 14th one will be studies on the production of sweet potato yogurt using selective probiotic bacteria. It is still a PhD work by one of our researchers. Thank you for listening.
Thank you. Thank you very much. I must say that having 14 proposed projects is a tall order. And I do hope that you will prioritize, because I've seen you have a number of anti-obesity studies, and none of those studies will take you less than 3 months. So even if they are running concurrently, having too many projects can, except you have very large teams. In any case, thank you very much for managing time, and we'll now take questions for you, please. So, we're running so much into the time for the next session, so we'll just take 3 questions, and you may wish to note down the questions and then address them. Okay, we have a retired director over there, one. In front of him, 2. Any question from that wing? 3? Oh my, so we end up with 4. Make them brief, please. Thank you very much.
Thank you, Mr. Madam Chairman. This is a very short question, it's just for my own perhaps interest. I was in Bujumbura some years ago, and I saw raw sweet potato being fed
Thank you. So I was very curious, and actually I wasn't permitted to ask many questions. I would have asked, do you just feed raw sweet potato and mix it what is their grinding whatever and make bread like that without perhaps grinding like we grind flour, cassava flour. It was just fed direct, and they said they don't even use sugar in it. Is it what was done here in this one that we saw? Second one is very short also. Before I went outside, my wife bought pink sweet potato. And no, the pink leaves. It has pink leaves. I was also very curious about it. Is it better nutritionally than the Mother's Delight that is, I don't know. Do you understand what I'm saying? So I want to know, because we already bought it, I want to know how good it is, how better the other one is than the Mother's Delight. Thank you.
Thank you very much. My name is Dr. Nonka. I want to commend the presenter. It's a very good presentation. But I just made some observations while you were presenting, especially this last one. I know that while we were here, the proposals used to have names of the researchers attached to them. I didn't observe any. Is it a new convention now? That's one. Secondly, when you were presenting the works done, I saw the one you mentioned that you go to the socioeconomics and you are doing it in food processing. Is that an agreement too? Even this last one now, there's one that involves machine, use of machine. That should have been in engineering also. Is there a collaboration we want to know? Thank you very much.
Thank you so much, Madam Chairman. My own is a fundamental one. We have been producing a lot of these value-added products over the years. And this time around, we are talking about commercialization. How far have you gone in commercializing them, rather than displaying them here every year? Eh? Some other people are trying to tap your technology, and they are trying to, claim one thing or the other. Yeah. What are you doing? Are they in the market? Eh? They are not. Eh? Please tell us how far you have gone to commercialize these things. Thank you.
So one of my questions is related to what he asked. If I go to ShopRite in the town, will I find your potato bread in the shop? That's one question. So second one, you discussed adding cinnamon into your products. So your cinnamon, does it come from you, or you are buying the cinnamon powder from somewhere? If you are buying it from India, I would like to sound a note of caution. There are no Indians here, I suppose. So in my lab, we get cinnamon from India and from Vietnam. And when we test those cinnamons, we find lead levels that are as high as 2,000 parts per billion. That is a very high level of lead in food that we consume by humans. So when you get your cinnamon, if you don't grow the plant yourself, if you import cinnamon from those countries, make sure you check the cinnamon for lead and cadmium and arsenic and mercury to be sure that your cinnamon is safe before you inject them into the food you are producing.
I don't know. So were you the person Okay, thank you very much. I was mixing it up.
Thank you very much. Dr. Sumoran has really asked a part of the question I have in mind. I noticed that the name of the program itself is Product Development Department also. is it not possible we incorporate and commercialization? Because it does appear that the name given to this program appears to be limiting them. They will only develop a product. What next after the development? The essence of the development is to make money. Today, when the economy is in their need of revitalization, I think the institute should look inwards and find a way of making money from most of their research findings. After developing, now you have, for so many years now, sweet potato bread has been on the shelf. This institute developed it over a long period of time. But if you go to the market, you don't get these things. I want to believe that farmers are not so free to pick some of these technologies because they may not know the know-how of how to go about it. So I will strongly recommend that, one, we change the name of the program, incorporate commercialization into it, and then empower them to, after developing these products, they should take one step further to commercialize so that they will be making some money from it. Thank you.
Thank you very much. The former director of that department wants to give us an information. So just give her the mic.
Thank you. I just want to start with Dr. Asamoah being one-time director in this institute. He should know better than commercialization is not anything you do with empty hand. You need money to do that. And the slogan here is that Egadro. Is that not true? Can I finish my question, my comment? That's another one. And again, we have extension department. Their duty is to go to all the departments in the institute, find out the innovations they have there, and extend to rural people. And not, you can't be doing research work in this and then involve yourself in commercialization. If you want to do commercialization, please start a new department that will be going there to take the innovation and commercialize it. You can be doing research and commercialization at the same time. Thank you very much.
Thank you very, very much. So we will now have the presenter take Dr. Joe. She will
answer that because she will answer all the technical questions, but the issue of change of name and commercialization is purely administrative, that she does not have the capacity to answer. And
definitely.
for commercialization, let me just tell you, the testimony, I'm going to give a testimony. Last year, they came and said what they are doing, that they need money to commercialize it. And I told the ED, we are not going to give them money, let's give them loan of 1 million naira, you will pay it back. The summary is that today, they have paid back, and they are operating, and they are even planning to buy a vehicle for the program that they will be using to distribute. So they are already working.
Thank you, my director.
Well, Dr. Joe, I still believe that saddling the researchers with this, I think the institute should be thinking of an industrial park at this time.
We have Umudike seed and we have the Umudike foods. We have it as part of the things that my executive director.
So that should be going to the business arm of the institute.
So and we already have a director that is in charge of that commercialization when it comes to Umudike foods. But we've started. I'm sure most of the people here have eaten our sweet potato bread. And most of our staff go home with our bread. Most of them don't buy from the local bakeries again. Yes. And we are proud of that product because it is fresh, it is natural. Yes. So I am proud that the product development program has taken it to that height, Ma. You need to visit our production units. We are already making steps even to secure NAFDAC numbers for all our products. We have started engaging NAFDAC now. Yes, ma'am. Okay, so I want to thank you for the interest our presentation has generated. Sir, you know, different food products have different ways of incorporating raw material. And it's also dependent on the end user. So for animal feed, it could be done like that. For this particular study on the bread, we use the puree. Why? Studies have shown that the puree is a better method of using the OFSP. A lot of work was done to check which form that is best to use for production of OFSP bread by SIP. So that has also been taken care of. So what we are doing, like I said, in product development, we develop recipe, we standardize the operation procedure, we are into quality check too. So you also asked about the purple one. This study that we took here gave me an insight in the advantages of having anthocyanin even over beta-carotene. So I said here that I am now telling the breeders that my program has found out that bread made from the OFSP, the purple flesh sweet potato, keeps longer. But texturally, sensorily, it's still not the best. It's hard. The color is not very appealing. We did not just with the instrument, we asked people. We gathered 20 people into the lab and they evaluated over 12 genotypes of that material, and it kept coming as an answer. I don't like the purple color. I don't like how hard it is. But when we went to check the shelf life, we found out that it can last up to 7 days with minimal growth on it. Okay. So, sir, you were asking why we didn't include authors in the presentation. It is a new policy. We want to give the proposals a fair chance. Let them come to the clearing room. To you people, you tell us which ones are good. If you see my name, you may be biased. So we want you to do it blindly, sir. So I think I've addressed the commercialization. I've told you that we started engaging NAFDAC, and we have also started.
Thank you, thank you. I think you've addressed all the questions. Please, can we clap for this woman? I think that the marketing department should also engage her. She will make a good marketer. Thank you very much. So we'll be moving on to, but madam, on a more serious note, 14 projects is too much. I think it's either you merge some of them or you face them and see what you can, so that you are able to have good results for the year. Thank you very much. So let's quickly move to the seed technology program. We're running behind time, so we're going to reduce the question time. And then I wish the presenters could be as fast as Madam Ihuono. Thank you very much. You have the floor, sir.
The chairman, the executive director, please permit me to stand on all the already established protocol. I'm here to present a work on seed technology program. So this is the outline, achievements, and lesson learned for work we did last year. That's the result. The outlook for the 25 cropping season, that's under the proposal. So this achievement we have weed management. Use of glyphosate at the rate of 4.0 kilogram unit per hectare with 6 sachets of adjuvant. Control weed effectively. 15 cm cassava length was the best planting length for both slant and horizontal cassava seed production. Use of potassium has played a complementary role in seed yam production, that's water yam, in soil moisture stress condition. And also 4 botanical, sativum, that's alum sativum, and alum indica, and curcumin longa, and zingiber officinalis, that is ginger. So, official extract can inhibit Pythium meritonium, causative agent of cocoyam root rot disease. And from the work, we have a evaluation of new herbicide formulations for weed control in cassava seed production in forest of Nigeria. You know that weeds are a serious problem to farmers. And some of the weeds are resistant to herbicide now. So we have to use adjuvant to help in enhancing the effectiveness of the herbicide. The objective is to assess different formulations of glyphosate and adjuvant for cost-effective weed management in cassava. So we have 9 treatment, and we also have glyphosate is active ingredient of the herbicide post we're having, and ammonium salt of glyphosate with adjuvant. This means that you have ammonium ions that help to inhibit the weed. So from the result, under weed density at 16 weeks after planting, treatment glyphosate with adjuvant 4.0 kg active plus 6 sachets of adjuvant control weed effectively with a less 0.9, and also that of weed biomass at 0.21. Now, in terms of weed control rating, that's under percentage weed rating. The same herbicide give us almost weed control rating of 10 to 100. Means that we don't have much mortality in our crop. So, from the result, the take home here is that use of glyphosate at rate of 4.0 kg active per hectare with 6 sachets of adjuvant control weed effectively. And the lesson learned here is that use of post-emergency herbicide is good for weed control in cassava seed production. The next trial is that evaluation of different methods for enhanced cassava seed multiplication. This one talking about seed multiplication, rapid multiplication of cassava. So the objective is to evaluate the best planting method of cassava varieties for seed stem production in Umudike, East Nigeria, and to assess the survival rate, that's under establishment, survival rate, growth, performance, and yield of cassava seed. Here we have the variety, Dixon, Game Changer, 871684, and TME 419. And the method we have here is the length is 5 cm, and we have horizontal planting 30 cm and slant planting 30 cm. So we found out that in terms of sprout count, 419 have a good percentage of survival in 2023 and 2024. Plant height also, TME have the highest of 184.1 for plant height meter, and also in 2023 and 2024. Number of leaves, 87184 has the highest leaf number, and also number of leaves both in both years. That is 2023 and 2024. And number of branches, the same treatment did very well. Now, take home here is that 15 cm cassava length, or the best planting length for both slant and horizontal planting for good cassava seeds. And let's learn here that slant and horizontal planting method are good for cassava seed production. Next, we have response to yield of some Nrcri alata variety under different moisture stress and potassium fertilization. We all know the role of potassium. Potassium helps in the bulkiness of the wood, especially of the tuber. This is a dry season trial. So we have a different available water depletion in percentage. We have 10%, 20% water depletion, and 30% and 40%. While the variety is Omoda 31 and
Just
Omoda
1 minute, please.
58,
Excuse
all
me.
are water, yeah.
Please, can we reduce the background noise from the table so that we don't distract the presenter, please? Thank you very much.
So we found that it showed that the percentage of water did not show any significant difference, but we still have seed yam and also seed yam and ware yams. You know, from the seed yam, you can still get the ware yam. Now, for the potassium help in the bulk of ware yam and that of seed yam. Take home here is potassium has played a complementary role for seed yam production in soil moisture stress. And the trial was able to produce encouraging seed yam yield, water yam in dry season under sprinkler irrigations. That's the method that we use to spray the water. Now, with the problem we have in cocoa yam, there's a disease called Pythium methodium and the first agent. So we are trying to use botanicals to control it. We are using chemical, but chemical is expensive. So we have to use botanicals to see how far it can inhibit this cocoyam root rot. So from the result, we found that alum sativum has almost 100% inhibition, followed by alum indica, and also ginger officinalis, that is ginger, perform very well, and also curcumin longa. 4 of them inhibit, when combined, inhibit the Pythium methodium causative agent. So the thing right here is that due to expensive chemical procurement by peasant farmers, plant extract can be used to substitute in the control of cocoyam root rot. So thank you for listening. We have 4 achievements for the year.
Thank you very much. You can go straight to your proposal and we can take everything together.
Now we have major. We have need for
technical team, please.
Proposal 1 has use of check of the cassava foundation seed. Okay, Nigeria. Objective to determine the use of this set in the multiplication of cassava seeds. 2. Evaluation of newly released cassava varieties for seed production at Nrcri UK. Objective to determine the suitability of these varieties for cassava seed production under field conditions. Certify the most productive varieties for large-scale cassava seed cultivation and distribution to farmers. Evaluation of the newly released yam varieties for seed production at Nrcri UK. Objective assess agronomic performance of newly released yam varieties for seed production. Identify varieties best suited for this yam provide recommendation for scaling up seed production and distribution to farmers effect of acceleration, identification, and control of microorganisms in CDM storage for enhancing quality and productivity of our CDM network. Objective to isolate and identify storage microorganisms associated with CDM in storage to evaluate the efficacy of different systems in controlling storage organisms, to evaluate the economic losses of shelf life caused by the storage organisms. Now, what is sanitary task for production aid and policy stability? We are select and define microorganisms of the seed and the sanitary practices used by farmers. Now, the project of the system plan for yam production using yam cells. Assess the demand for yam seed and identify target customers. Apply the production process, including source selection, field test and planting, as well as crop assessment and surveillance. To determine the prospect of working and training for the yam seed production. The growth marketing strategy and sales and potential. Now, this is organic amendment combined with oil fertilizer in the production of food oil. Objective to assess the use attributes of the amendment, to determine the physical and chemical properties of the soil, to determine the soil microbial populations of the different treatments, and to evaluate the economic analysis. 9. Design and fabrication of aeroponic system for CDM production. This is an engineering work. To design and fabricate an aeroponic system of 1.6 by 0.8 meters for seed production. To improve the production of healthy seeds. To introduce modern innovation in seed production. 10. Sustainable quality. Quality and productivity, to assess the impact of organic media on soil physical, chemical, and biological properties, to determine the optimal ratio of organic media to cassava growth, and to investigate the effect of organic media on cassava disease and pest management. Now 11, use of big set of seed yam, using to see under yam in set. So use of big set of seed yam in the multiplication of foundation seed at Umudike. To determine the use of dig set in the multiplication of seed yams. To determine the availability of planting materials, especially at the beginning of the cropping season. To determine the quality and viability yam seed to farmers. Thanks for listening.
Thank you very much. We have listened to the report for the year and also the proposed activities for the coming year. So we'll now take questions for the seed technology program. I see that the only project you have in aeroponics is the engineering. So who are you collaborating with? I'm not sure the Institute has an agric engineering unit. Eh? Okay, okay. Thank you very much. So please, questions for him. This time I will now be taking the questions bit by bit. So any question from this segment? From the middle, okay. Dr. Ano, okay. Okay, sir. So those two, please. I think we're running out of time.
Thank you for the beautiful presentation. My question is on effect of potassium on water stress management in yam production. You did not give us the nutrient status of the soil. You did not give us the amount of the other nutrients you added, nitrogen and phosphorus and maybe other ones. Then from what you presented, half, I suppose is the half of the recommended dose, and then 1 and 1.5. If you check 1 and 1.5, they are the same. So I don't understand you concluded that potassium is very important in water management, but the recommended rate of potassium is what gave the highest yield. So I can't understand the usefulness of the experiment. Thank you.
The next question, please. Over there.
Thank you so much. Dr. Musa is my name. When you're making recommendations for the planting length of cassava stick, are you aware of the fact that you can have 15 centimeter length with about only 2 nodes? Are you aware of the fact that you can have 15 centimeter length with about 20 nodes? So the emphasis should not be on the length, but number of nodes and the planting material. Thank you.
Okay, thank you. That should be the last, please.
Yeah, thank you very much. Mine is a general question. There are new information available from the work done recently, perhaps in the last 1 year. But through power presentations, we are not able to have anything to take away for purposes of studying them or reading them and internalizing the information. So I don't know what is going to happen. I know it may not be possible with all this deluge of research findings to say you send them online or maybe in soft copy. I don't know how we can, how do we cope with this situation? For example, there are some new information that I have observed from the presentations on cassava seeds and so on, ratooning and all that kind of thing. Yeah, some of them, we need to use them. But before you know it, it comes as a flash and it goes. And there's no way we can internalize all of them immediately. I don't know how this can be done, how this problem can be solved. Thank you.
Thank you very much. I don't think that question will go to him. Acting ED, sir. From what, sir, you did not introduce yourself. Yes, sir.
Okay, Professor Jude Njoko. I'm Professor Jude Njoko.
I know, for the audience, sir. Okay. Thank you very much. Acting ED, sir, I think that he has raised a very important issue going forward that the planning may have to consider in future in-house. We can have a synopsis of recommendations coming from the research reports, because the synopsis can just come as small papers that can go around or that interested people can pick from the registration desk. Because the issue with the e-age we are in now is that the informations come so fast, and you may not have a platform where everybody here can get them, but we can still have just pamphlets that summarize recommendations, which is what people will need to take away. Thank you very much. So you can take the 2 questions now. Professor Jude, does that address your concerns. Thank you very much.
Thank you very much for the question and comment. Dr. Ano, sir, with this soil analysis, this show is in the annual report. The annual report that they are going to present now is there. We did it to know the nutrient status of the soil before the application of the potassium rate, the 0, 1, 1.5, and so that's the food dosage.
What
So
he's trying to say is that the difference between 1 and 1.5 is too narrow. Am I correct? Thank you very much.
That's what the results gave us. And it worked. If they are the same, but there's similarity on it, that we see, there you see a difference, but it's not a significant there. But you see that there's a similarity on it. Noted. Okay, another one is 15 cm, the note. You know, have 25 cm, we have 30 cm, and we have 15 cm. The new intake there is 15 cm. That's the length we are looking for, not the note.
Thank you very much. Please, can we clap for Dr. Koriocha for the report, the proposal, and the questions. Thank you very much. So let's have the Genetic Resource Program team. The Genetic Resource Program team, you don't have a report. You just have a proposal and questions. Is it a new program? Okay, thank you very much. I needed to confirm that.
Chairperson, Ma, Executive Director, permit me to stand on the already established protocol. The Genetic Resources Program in Nrcri is now baby program of the Institute. So, this is our broad objective, to assemble and conserve crop variants of fruit and tuber crops of economic importance in Nigeria. To achieve this, objective, okay, go back. The other one. Yes. To achieve this objective, we organized exploration and collection of wild and domesticated species of root and tuber crops across Nigeria. Conservation of endangered wild and domesticated species of fruit and tuber crops for future use. Make available germplasm for exchange, breeding, and other uses. This is all we have in our genetic resources program. We have cassava, we have, the number is 236 accessions. Sweet potato, we have 16 accessions. Yam, we have 144 accessions. Sweet potato, we have 20 accessions. And ginger, we have 2 accessions. Turmeric, we have 21. African yam bean, we have 36. In our taro, we have 6. In our tenia, we have 3. These units have been elevated to a program due to dedicated staff and hard work. The program has been able to conserve the genetic resources, that's the germplasm stroke, the gene bank of the institute in the following mandate crops. These are the mandate crops we have. This is all we have in their number. Yam, cassava. We have, I have mentioned them. Cassava is 236, yam is 144. Taro is 6. Tenya is 3. Then we have a potato 20, we have turmeric 21. We have Polystonia arrowroot 1, we have ginger 2, we have African yam bean 36. Then the new proposal. Our aim is just for exploration, collection, maintenance, and conservation. That's the aim of the genetic resources program. Then our new these are the people that are working in the program. Then our new proposal is based characterization and evaluation of fruit and tuber crops accession of plant genetic resources in Nrcri and Umudike. The first one we have to do is collection. We do collection of root and tuber crops accessions in Nrcri and Umudike. Then the objectives. Something is missing. For activity 2, the other one is activity 1. Activity 2 is characterization and evaluation of root and tuber crops, accessions of plant genetic resources in Nrcri and Umudike. Our objective, characterization and evaluation, morphological and molecular characterization of germplasm and screening for key agronomic traits. Then when we talk of the traits, we say drought, tolerance, pest tolerance, and others. To identify superior genotypes for key traits such as yield, pest and disease resistance, and the drought tolerance. To preserve valuable germplasm for crop improvements. Then this activity 3, conservation and documentation of fruit and tuber crops accessions in the genetic resources in Umudike. Then the objective, to conserve and document diverse root and tuber crops accessions from various agroecological zones in Nigeria, both in situ and ex situ, using modern techniques, gene banks, and tissue culture, to document and preserve valuable germplasm for future crop improvement program, to ensure the availability of the conserved materials for research, breeding, and production. Then this one is the mobile app based digital passport system for genetic resources data management. The objective, developing a centralized mobile accessible database for germplasm passport information, implementing a QRD code and FID code based tracking for quick identification and traceability, enabling seamless integration with phenotypic and genotypic based databases for threat selection, facilitating secured data sharing and collection between breeding institutions, incorporating the LD-driven analytics to assist breeders in germplasm selection. This is all we have for the proposal.
Can we clap for her? Well, it's a new program and we have heard proposals. Just to note that I think it's AI driven there, not AL. The last point you made, I believe you will be having a lot of collaboration to achieve the things you have proposed. Do we have questions or inputs to make into these proposals? So, do we have any inputs or suggestions? Okay, sir. 1. Okay, any other person? 2, 3. 4, the last, please, so that we can give, can step aside for the next session.
At Ibuku, we found out that the major issue that determines yield goes directly to the gene. We have tried several experiments on that. So I think our lead person is going to make a presentation and we take it from there. So your own department and your own program is very, very key to what we are saying. Not the best way to have the best seed for each crop, it goes down to the gene. So if we have to work more on the gene, selecting the gene that makes the best yield. So I give you kudos for that. Please, thank you.
Question or contribution?
Yeah. Mine is just a comment. I don't know the relationship between you. I hope you are linking properly with the in vitro gene bank conservation of these materials, because I can see that, for instance, sweet potato. In 1990, I was at IITA. I received well over 100 sections of sweet potato. And by the time I was coming back, I was leaving the program in 2000, that is plant tissue culture, in 2008. I lost only one section out of the 100 and something I had. That is sweet potato. And we had quite a lot of also ginger beyond the two that we are now dangling. We had Rio de Janeiro, Waida Loka, Himachal Pradesh, all of them were there, in vitro, in the tissue culture. And the idea is, if you lose materials in the field because of vagaries of this thing, you should fall back on this thing and then recapture it. I don't know whether you have a proper link with them to ensure that there's no genetic erosion. Thank you.
Oh, thank you, Mr. Chairman and the presenter. Thank you again for a wonderful presentation. But I have always been worried since the beginning of the presentations, particularly when people are making reports on turmeric. And the question that has been bothering me is which turmeric? Because there are principally 3 types of turmeric. Which turmeric are you talking about? Are you talking about the Xanthorrhiza? Or are you talking about the white turmeric or the yellow turmeric? That's my question. And I think this is the right program to address that.
Thank you very much. Please, for information, I want to know of what is the economic importance of the African yam bean? You mentioned among the things you are assessing. Thank you.
Yeah, thank you so much. Just to buttress what my colleague has asked or commented on, our major problem here is on genetic erosion. You have so many this year, next year the number will go down, but there is no proposal on conservation. I was thinking that it should be a major issue in the program. Thank you.
So thank you very much. We've heard the questions. I was about to also raise the issue of conservation. So Dr. Ano has taken it up from there. So please, you can then address the questions, because the contributions are well taken. Okay? And then also tell us if you plan to have an activity linked to conservation.
Thank you for your observation. The conservation is on activity 1. I didn't say it here. So, but in our ward is there. Our conservation work is on activity 1. But for the man from Anambra, thank you for your for what you noticed, because you said that without gene, that nothing will be achieved. Even yield and pest resistance. Thank you for your observation. Then for tissue culture and what Dr. Chukwu observed. You have been here before. You know how we are managing both our institute and our exit. So that one is a management issue, but we are trying. For turmeric, we have 21 accessions. We have 21 accessions, even up to tomorrow. We plant them, we conserve them, even in our store is there. So the accessions are still 21 for turmeric now. Then for African yam bean, we have it in our, as one of our districts. African yam bean is one of the things we conserve in our genetic resources, program because of its value.
Thank you very much. Please, can we clap for her? So with this, we have come to the end of this technical session. I want to thank all the presenters for good presentations and to comment that it's getting better each year. I also want to thank the audience for active participation, good contributions, thought-provoking questions, and of course the technical crew for ensuring that we didn't have too many time outs. Thank you very much. So we will step aside. Sam.
Please, can we give our chairperson a wonderful round of applause? Her team has really done justice to this very section, and we want to really appreciate them. Thank you, Prof, for that wonderful delivery. We want to move on to the next lead paper. This should be the third lead paper for this event. And it's going to be personal perspective of yam cultivation. And to handle it will be Professor Uchenna Nwosu. And I know he's ready for us. But before we invite Prof. Nwosu, we want to welcome the chairman of this very session. And he is Professor Emmanuel Nzebula, the Professor of Environmental Management, Michael Okpara, University of Agriculture, Mudike. Okay, I thought I cited him. Yeah, maybe we'll call on him. But then let the rapporteurs please come to the high table. Dr. Okezie Chukwemeka and Mrs. Juliet Nrcri, please can they come to the high table? Yeah. Let me crave our indulgence and to quickly request that retired director, Dr. J.E.G. Kogo, should please chair this particular session. Dr. Kogo, sir. Can we put our hands together for Dr. Kogo? Yes, yes. Please, can we clap for Dr. Kogo? Unless if you are telling me that your smiles can be taken for the applauds, then I will take it. If not, because you're smiling too much and you're not clapping, can we clap and celebrate Dr. Kogo? At this point in time, I want to also welcome to the high table our presenter, Professor Uchenna Nwosu. Please, can we welcome Professor Nwosu to the high table? Okay.
Prof. Ndeo.
Chairman, sir, over to you.
Thank you very much. I was saying that I thought that if I'm in the panel, I should not be as the chairman, but since it is allowed, I want to thank you for granting me this opportunity of sharing this session. And I believe as we did well in the last two presentations and two lead papers, this is the third one. I believe that you're going to also cooperate with us here so that we'll do well. Having said that, okay, I would like to invite Prof. I don't know whether he needs introduction because he has been here before. If not, because of time. All right. Okay. Because Professor Nwosu is a medical doctor turned we say a farmer, not even a farmer, a researcher. Let me just stop there because of time. We are going to listen to him. So, sir, you are welcome, and I would like to turn over the podium to you to present your lead paper 3. Thank you. Can you give me a round of applause while he goes to...
Say thank you to Dr. Casey Mado for inviting me here to, I want to say big thank you to Dr. Casey Mado for inviting me here today to give my personal perspective on yam cultivation. I also like to thank the ED, or the acting ED, as I heard it said. I want to also give my thanks to Dr. Mary Maza, with whom I had been this morning, and of course to Dr. Johnny Kogo, with whom the research organization at Ibugu is named with. The YAM Research Information Center at Ibuku. Now, the first slide is a 2-minute, very short slide. I don't know whether it will play or not. I don't see it here, the first slide. Does this play? Because this slide is really about how I got
my epiphany into yam cultivation. I'll be very happy if it can play. If not, I'll move on. I can move on. Okay. All right. Since I did not play that video, I can tell you what it's all about. 2 years ago, 3 years ago, well, I've had heard about planting yam in bags. And it was started by one Chief Sonny Okafor of Omunachi in 2018. I saw a video of it. And 2 years later, in 2020, my cook had a piece of yam head that he was going to discard. Yam head with very little flesh. He was going to throw away. And I said to him, because of the video I had seen, let me plant it. When I planted this yam head, when I harvested it, it gave me a big yam. And I wish the video was on. And that stimulated my interest, because I had always thought that when you plant, if you want a big yam, you plant a big yam. And here I was planting a yam with hardly any flesh, and it gave me a big yam. And because it stimulated my interest, and I was wondering what caused this. Why is this being it's the video that's playing. Okay, where is the sound? There's no sound. Okay. All right, just view the video. That's my yam epiphany. Oh, okay. Forget what I did not know that this will be ever seen in public. April, not June. Last week of April. Okay, so that was how my interest in yam started. That was in 2021. Because I had to speculate what caused this yam to be so big. And that was where my thoughts went into genetics. I felt that there must exist a gene for growth gene in the yam. And that was what Chief Okafor was referring to. It was a speculation. We have not found any such things. But I speculated there had to be a gene for that. Now, there was a slide before that, no? Okay. So, what I did, being a perinatologist, I was trained in high-risk obstetrics in the United States. I had always been, once I began to think of yam, I decided to plant and see what happens. Now, what I heard from my parents and what I see in some yam literature is that when you plant yam, it rots. And so the first thing I did was just to do what I called sproutology. Just like we have in medicine, we have embryology. What happens when you plant yam? And please forgive me if I'm just repeating what is obvious. But what I did then was to plant yam in bag and use it as a research tool. So what I would do was from various days, I would dig it out and see what was happening. And so what I found here is that when you plant yam, it doesn't rot. First thing it does, it sends out, the yam tuber itself sends out roots. And what I did was to unearth the same tuber from day to day, at different days. This one you're looking at here was uprooted at 20 days. The next one, right? Which uproot? Down. Where's 30? Yeah. Is it this? When I say next to you, okay. Yeah. Okay. Yeah. So this one is at 31 days, and you can begin to see a sprout right there. We go to the next one. This one is 35 days. The sprout is showing a lot more clearly. This one is okay, first bud in early bud starting. This one is at 39 days. At that point, you see the real roots. All these were what was feeding, what was really happening to whatever autolysis that was going on inside the yam and liberating all kinds of chemicals, enzymes, and so forth. But now you first see the first roots. There are no root hairs. All the root hairs are in this one. So these are just the starting of roots. On day 39, where do I point this? What do I point it to, sir? Okay. All right, 50 days. So at that point, you can see the real roots of the yam are beginning to be established. But the adventitious roots with root hairs and so that are really feeding the process are still in charge. Now, excuse me. This one shows a yam that we dug up even at harvesting, and they found that even the parenchyma of the mother yam, I call it mother yam, was still alive. What I had shown you before was using yam set, awaji as we call it in Igbo, bang. But using a whole yam, you see the same activity. So, excuse me. So what I can deduce here is that this was only on the 9th day, but this degree was not reached with Awaji until like day 69. So that tells me that the full yam is faster in development. Now, the next thing I'll just tell you next. Thank you.
Is it okay? Somebody can operate it for you.
Go way back. Okay, right. Thank you. Let's see that. Yeah, the first, the one we missed. Yamin Park. Okay, go down. No, let's forget that. Next. Oh, okay.
What's the problem?
Okay. So, what I then did, when I say my experiences, I then followed the growth of my yam, and there were things that came up there that I want to share with you. One of them should be on the next slide. There's no write-up. One of them should be on the next slide. Where's the next slide? This has to do with the flowers of the yam that I encountered.
What is happening? Is anybody helping to operate the video?
I have a flash. Maybe you can use. Okay. Okay. Thank you. Okay. This graph was taken from a book from Dr. Onweme, I.C. Onweme, who some of you know. The interesting thing about this graph is that it is from zero, it is this portion that is not on the tuber growth that I demonstrated. Professor Onweme is my friend. He was my schoolmate. Thank you very much. We both were colleagues at University of Ife, and we were made professors on the same day in 1980. He moved.
Please, please. Let's listen to him. We don't have much time remaining, please.
Thereafter, he moved to Futa to become the dean of the Nrcri, while I relocated to Ibuku to start my rural hospital. Now, this is what I found. And I had always been seeing it. I didn't know what it was. I was told that it was flour. Does anybody know what it is? Is it in any book? Okay, next slide, please. These are mature flowers. Next slide. You have it now. Okay, this is when it is dried. Next slide, please. Okay. What I did was enlarge it. And this is what I saw. It was beginning to look like flowers. Next slide, please. Next slide. Okay. Yeah. Eventually, we got to this. This is that little flower from the spikes you are looking at. I sectioned it and got this picture, and I convinced myself that it's indeed a flower. You can see the stalk, you can see the corolla, you can see the calyx, and so forth. So again, like I said, what I'm showing you is my experiences, what I encountered. I was looking at from as a scientist. Next slide, please. Yeah, well, and then when I looked at for the literature, trying to find this flower, I didn't. All I found was this one from Discoria borbifera. I heard you say ado. This is the flower for it. That was the only flower I could find in yam literature. Next slide, please. Then next thing I found was galls. I saw these things on the leaves. Because what do you call it? Is that gall, really? Okay. I didn't know what it was, and then I decided to investigate. Next slide, please. Yeah, I sliced it in a transverse section, and I found those tubes over there at the center. Next slide, please. And I sliced it in a sagittal section and saw those tubes ejecting conidium. Okay? So I convinced myself that this was fungus. The fungus is in the group of Coletotrichum. And this was the mycelium. At different slices, I was getting different parts of the fungus. Next slide, please. Next slide. Here is the fungus ejecting the conidia, which was what really the sexual spores that spread and infect more leaves. Next slide, please. This is the one that has ejected all the conidia and is drying off. This is the old part of old fungus. Next slide, please. Next. Oh, it's gone back to the number one. Okay, good. Next, yeah. Yeah. Yes, hold it there. Yes, this is an interesting observation I made. This was a yam I planted in a bag in a shade. And the vine was so tiny, I was expecting to dig out a tiny yam. And look what I got from that. From a you can look at the tiny vine and look at the product from a shade. Next slide, please. By the way, let me just say that also at that time I also planted yams in bright sunlight, and some of them had very thick vines. The proximal vines were very thick, and when I harvested them, I got buffalo chua. That is very small product. So this is my little attempt at questioning the role of the nature of the soil in the yam. What I did was, this is, like I said, it's just a little experiment in which only 6 bags were involved in each case. I planted some yams, 6 yams, in sharp sand in the bag. Next was in plaster sand. Plaster sand is sand that has maybe a little bit humus. Plaster, it's not sharp sand. The next one was planted in topsoil, and the next one was topsoil with manure, and the next one was topsoil with manure and fertilizer. And Oh, play that. Wait, forget about playing. It's something. No, go back now, please, to the result. This is what I got in terms of the yield from those experiments. The first one was the topsoil, and the right writings are the propagational index. What I call propagational index is when I divide the planting weight by the yielding weight. It tells me how many times the planted thing multiplied itself, and that's the propagational index. And the propagational index for the topsoil was 4. Okay, so the point was that I did in fact get some yam, the propagational index from from sharp sand was 1.7. Next slide, please. This is the problem we have with the bags. The roots form a cast, and they don't escape. Next slide. Next slide. Same thing. Next slide. Next slide. Yeah, this form of the way this yam has roots has to do with what I'm going to show you next slide. Next slide. What one Reverend Father, Philip Okoye, discovered is that you can plant yam in a pit that is about 3 feet by 3 feet deep, and you get big yams. Next yam, please. Next slide. And this is the kind of yam you get from that. Next slide. These yams are carried by 2 people, planted in a pit, 3 feet by 3 feet deep. Next slide, please. This is the one I myself got last year from such a plant. And this wasn't the biggest. Next slide, please. This is the kind of yam you get. It's not the manure that is doing it. It is the easy penetrability of the soil. Next yam, please. This is one that was used for Thanksgiving. I mean, for Igwansi. That is, next slide. Okay, so now one of those yams weighed 160 kg. 160 kg, which means that it was producing, let me see, you are doing 2 kgs per day, 2 kilograms per day, or 1.3 kilograms per second. Next slide. Now, my only difference with with Reverend Father Okoye is that he thinks that it's photosynthesis that is causing yam growth. I mean, this kind of yam yield. Even though he's getting things that is obvious, he speaks for himself that photosynthesis is not what caused it. Now, the reason I put this slide here is Dr. Okoye does everything to make sure that the yam has sun exposure to improve photosynthesis. But then I've seen this kind of yam planted in the north, some parts in Benue, where they are not even staked at all. And they give bigger yams than the ones that are staked. Next slide, please. Next slide. Next slide. So, okay, forget that then. I'm not going to deal with all those things. What I am here to say to you is that what controls yam growth is not photosynthesis only. In fact, it does a minimum. Photosynthesis produces glucose that nourishes the yam plant itself, the tuber, the leaves. It doesn't make excess yam sugar that is piped down through the phloem that very narrow things to store at the rate of 1 gram and a second. Okay, so next slide, please. I want to show you my own take on yam yield. My take on yam yield is that the ease of soil penetrability is 60%. This is my own perspective. Photosynthesis is probably 15%. Yam DNA, probably 15%. Soil manure, probably 10%. And fertilizer, 0. And I just want to close with this.
I thought that ovation can be translated into clapping. Thank you so much, Prof. You can take your seat. Yeah. At this point in time, ladies and gentlemen, you will agree with me that Prof has done justice to this title, sharing his own perspective to the issue of yam cultivation. And without much ado, we want to welcome the panelists swiftly. And this session will be anchored by Dr. B.C. Okoye. He will moderate this very session. And we want to welcome Dr. J.G. Ikogo.
I'm already here.
Yes.
Welcome.
Yes, please, sir. Okay. Yeah. One of our retired directors and former coordinator of YAM program. We also want to welcome Dr. Innocent Onyekwere. Dr. Innocent Onyekwere is an assistant director and the coordinator of Farming System Research Program. Please, can you join the panelists, Dr. Innocent? Please, let's applaud them as they come. We also have Chief Sir Christopher Wanebu. I cited him yesterday. Okay, okay. Sir, please, can you join the panelists? Let's applaud him. Also, Mr. Uche Onoha, the Secretary of AFAN. Is he here? Okay, he will join the panelists. And then we have also a lady to balance the gender dimension. And so we have Dr. Joy Emerald. She is the head Nrcri Climate Adaptation Agriculture. Okay, Dr. Emerald is coming. Yes, sir. Please, Dr. Koye.
Yeah, thank you very much. Thank you for that wonderful presentation done for yamming bags. So we want to address issues that's likely to arise from the paper presented. And we will take it as fast as possible. We will start with Dr. Joy Emerald. From the presentation and from your own experience in plant physiology, what do you think are your views on the ranking of factors contributing to yam yield by the presenter?
Thank you very much. That's the last slide, and first of all, I want to thank the presenter because all he spoke was based on his personal perspective. But I'm going to talk more of scientific perspective of what we've done in the Institute and the work that's been done in yam across the world and other yam producing regions in the world. I tend to agree about the ranking from soil penetrability to fertilizers. Yeah, but I don't agree with the rates of percentage allocated to them. And I have my reasons. For fertilizer, I agree about that, zero contribution. And I will come to that. When you talk about inorganic fertilizer, we've been working on this for the past 2 years, looking at various works and even the works done here. We've not been able to get work that outrightly addressed it and be able to transfer or translocate the assimilates into and translocate the assimilates into the storage zone, which is the root, which is the tuber. So I may stop here because of time, but that is what I am saying, that scientifically, without photosynthesis, there will be nothing like food production to be transferred to the storage organ, that is the tuber. Thank you very much.
Thank you very much. Thank you very much, Dr. Emerald. Sorry I cut you short. You know, it's a semester workload, and we must address issues immediately and end today. Let's hear from the agronomist, Dr. Ikog. Maybe in 60 seconds, do you feel there are specific agronomic strategies or techniques that can help prevent or address challenges in urban farming? Because the yam in bags is being proposed for peri-urban agriculture. So what do you think are agronomic issues that can come up to promote this?
I think what the Prof mentioned from his experiment, the soil where you plant these materials is important. That was perhaps why he used sandy soil, this soil, this soil. And if you look at recommendations for planting crops like you make sure that soil is humus, or at least something that the tuber can push down into the soil. That and sand is sand will just have to go and the plant will not be able to get it. So it's a mixture. It will make it difficult for the tuber to push in. So it has to be soil that is loose. Let me use the common language so that the tuberization will be such that it will push down. And that is why if you plant it in bags, if the soil in the bag is loose, you see that you get a big yam that is long. If it is not, if it hits a hard pan, it will swell and it will be wide. So, agronomically, therefore, soil where you plant it is very, very important. And we advise those in town, of course, I do it, I plant even in town here in bags. You have to select the type of soil. You have to go and make sure you get the soil in the neighborhood that is loamy or that you know that is manure, where you throw, they have been throwing away things, not newly, one that has formed good humus, so that you mix it and it will give you good yield of soil, of yam that is whole. It's not patchy. There are some that you know that the soil was good and it was trying to find its way to tuberize. Let me stop there.
Thank you very much, sir. Yeah, nice analogy. Dr. Nrcri, you are coming from a farming systems perspective now. Intensification or extensification, what do you think this approach integrating with the farming systems, please?
Yeah, thank you very much, Mr. Moderator. So I want to thank Professor Nwusu for a job well done and the intensive research work on this aspect. In farming systems, we call this approach systems farming system. The space farming systems, because it will be good for people in urban areas, it will be good for people that don't have land. Let's look at our present population. Though Nigeria is ranking #1 in yam production, and at the same time in land subjected to yam production. So we are cultivating about 48 to 61 metric tons, which is about 60 to 70 of the global population. And we look at the yield. When you come in terms of a white yam, we have a yield of about 10 to 15 tons per hectare, whereas our water yam will give us 16 to 20 tons per hectare, but cultivated to a land of 2.9 million hectares. You are saying land of 2.9 million hectares. What will happen in the future? Let's say about 2010. We are having population explosion, urbanization, we are building, all this land will reduce. The best thing for us is this thing we call the space farming systems, whereby we can cultivate our yams using bags. Because if you cultivate your yam using bags, when you plant on that land, now you are going to have 10,000 stands by 1 meter apart. But when you use bags, you are going to have 20,000 stands. So if we can get yield of 10 to 15 tons of our white yam, so if we use bags, know right now that we are going to have up to 20 to about 30 tons per hectare. And it will go a long way. Instead of that 48 to 61 tons per hectare annually, we may end up producing 70 tons or over that annually. And it will help in terms of solving the problem of food security. Even if you come in terms of production, even in terms of consumption, Nigeria is still #1. So we need to intensify using this system.
Yeah, thank you very much. Thank you very much. Chief Nwanebu, this system is different from the traditional method of yam cultivation we used to know. In terms of drudgery, labor, what do you think? Are we there? Is it better? Scooping soil in sandbags? Does it take more time?
Thank you. In his explanations, he applied both the ancient method of yam production and the modern method, which he explained is planting in bags. Now, my colleague has explained that planting in bags is likely going to save us land as we have population explosion in view. But let us also remember that each time we are planting in bags, we are scooping the sand from the land. And what we are removing is the topsoil and the humus soil from that land. And over a period of time, we are going to devastate that land. It will no longer be useful for agricultural production. And what we are asking is, if we continue in this form, are we not going to come at a time where there will be difficulty of getting the sand, the soil, on which we are going to use in planting yam in bags? And then are we not going to create a situation where the very present labor in use will become very lousy in actually going to the farm to cultivate yam? Then, let me further explain on what you have asked, that the labor-intensive, the labor involvement in scooping sand into the bag and planting yam inside the bag is more involving than when you go directly on the land. Because immediately you're able to clear the land, the next thing you go into is the land preparation, which you might use by mechanical method or human labor. But in this respect, you will go to where you have a good sand or soil, the humus aspect of it and the rest of them, scoop it from there and carry it to the place where you want to plant. So it's almost a double labor involvement. And again, let us remember also that after having used the sand scooped into the bag for the first time, he did not tell us that he reused it again in the next farming season, which means after the first use of the sand, it has become a waste. And then for how long shall we go into this practice and succeed in producing the food that we are looking forward for? Thank you.
Thank you very much. Thank you very much for this exposition. Yeah. We are rubbing minds. Mr. Uche Onoha, you are the chairman of All Farmers Association. Do you think this system is sustainable? Some literature has described smallholding as farmers with 1-2 hectares. How sustainable is this? Can you envisage yam in bags in 1 hectare? We're talking of food security in this country. Is it sustainable?
Yeah. Good morning, all. Yeah, thank you for having me. I'm actually the secretary of farmers in Abia State. I believe yam cultivation in bags is a very good option for some of us because I'm also into yam production and I'm doing both on the farmland and in bags. And the advantage is that land